All right. Well, thanks everybody. We have a great internal speaker today. So coming to the end of the year, and we'll have some home cooking here for the next couple of weeks. So today we're lucky enough to have Dr. Kathleen Martin, who's a PhD within the section of cardiovascular medicine. And she's going to be joining us to talk about some really exciting research that she's been a part of. So just to give you a little bit of background about who she is, she graduated from Hiram College, where she majored in biology, earned her PhD at Case Western Reserve in physiology and biophysics, and completed postdoctoral fellowships in cell biology at Harvard Medical School. She then joined the faculty at Dartmouth in 2000 in the section of vascular surgery and Department of Pharmacology, where she initiated her research program studying vascular smooth muscle cell biology and cardiovascular disease. In 2009, she became an associate professor of medicine and pharmacology at Yale and became a full professor in 2019. She has held multiple leadership positions with the AHA, has served as an NIH Charter Study Section member, and currently serves as the co-director of the Yale Cardiovascular Research Center. Dr. Martin's research lab aims to define cellular signaling and epigenetic mechanisms that regulate vascular smooth muscle cell function in intimal hyperplasia, atherosclerosis, and transplant vasculopathy, which is why we have her here today. So without any further ado, I will turn this over to Dr. Martin, and thank you again for so much for taking the time to join us today.
Well, thank you for that very kind introduction and for inviting me to speak with you today. I'm excited to tell you about some of our recent work on smooth muscle cells in transplant vasculopathy and implications for preventive strategies. So just a quick overview of what we'll cover: smooth muscle cells, a little introduction to what makes them so unique. And spoiler alert, it's that they have a remarkable plasticity. And I'll tell you about some of the epigenetic mechanisms that we think are responsible for that. And then what does this all mean for transplant vasculopathy? I'll show you some data from mouse models, some clinical data, and what this may mean for prevention strategies. So smooth muscle cells, as you all know, make up the muscular layer of blood vessels, and their ability to contract and relax allows for regulation of blood flow and blood pressure. And their remarkable plasticity allows for growth, remodeling, and repair of the vasculature. But inappropriate vascular remodeling contributes to many cardiovascular diseases. And these include atherosclerosis, where smooth muscle cells not only make up the protective fibrous cap, but also contribute to the interior of the lesion. Intimal hyperplasia, where smooth muscle cells form these neointimal lesions after angioplasty or surgical interventions, aneurysms, and allograft vasculopathy, which we'll be talking about today. This is an example. This is a rodent model of balloon angioplasty, and you can see this thick, this dark red layer is the smooth muscle cell layer in a normal healthy vessel. And in response to mechanical injury caused by the balloon and the catheter and the hyperinflation, the smooth muscle cells go into repair mode. They become highly proliferative and they migrate into the lumen of the vessel, and they can form these occlusive lesions. And when this happens, the smooth muscle cells are not only proliferating and migrating, they're also de-differentiating. They quite dramatically change their gene expression and their morphology, their whole phenotype. They actually change into fibroblast type cells. And this is an example of a cell culture model of this dramatic plasticity of smooth muscle cells. Here you can see the mature differentiated phenotype. The myocyte phenotype is elongated. It has this characteristic very thin spindle shape, and they express a repertoire of smooth muscle specific contractile proteins that allow them to contract and relax. But when they go into this repair mode in response to injury, they look completely different. You can see they now have lost that beautiful myocyte morphology, and they have this flattened, enlarged rhomboid morphology that's characteristic of fibroblasts. And we often call this the synthetic phenotype because instead of expressing contractile proteins, now they express a lot of extracellular matrix proteins like collagen and fibronectin to help repair So they proliferate, migrate, have high rates of protein synthesis and this fibroblast morphology. But interestingly, this transition is reversible. They can differentiate back to the healthy myocyte phenotype as well. And this plasticity is very unusual because most cells, when they differentiate and become a specialized cell, they undergo what we call terminal differentiation. For example, once a stem cell becomes a skeletal muscle cell, it will never assume any identity other than a skeletal muscle cell. It will also never proliferate again. But smooth muscles are very different. Smooth muscle cells can not only take on this fibroblast morphology, they can actually switch and become macrophage-like foam cells, which we've learned only in recent years from genetic lineage tracing, that many of the macrophages in atherosclerotic lesions are actually derived from smooth muscle cells. Smooth muscle cells can also become osteochondrocytes, bone-forming cells that contribute to calcification in blood vessels and atherosclerotic plaques. And in rare circumstances, they can even become adipocytes. So how is it possible that smooth muscle cells have this plasticity to become other cell types with such dramatic changes in gene expression? My lab is interested, we're very focused on the idea that epigenetic reprogramming allows smooth muscle cells to have access to all these very different genetic programs. And here's an example of how we think this works. So I mentioned skeletal muscle cells, for example, when they terminally differentiate, they still have all of the genes in the human genome. They have all 23 chromosome pairs, but they no longer need to express the genes found in blood cells or neurons or cardiac myocytes. So all of these gene programs are packed away in what is called heterochromatin, this tightly compressed, condensed DNA that's found at the periphery of the nucleus. So in this way, it allows them to pack away all these genes that aren't necessary, whereas the genes that are active and being expressed in that particular cell type are found in open chromatin or euchromatin. This chromatin looks very different in that the nucleosomes are spaced far apart so that the transcription factors and the polymerases can get in there. So epigenetic mechanisms are what regulates how DNA is packaged. These can be post-translational modifications of the histone proteins. So these proteins form a core, like a spool of thread around which the DNA is wrapped. And these spools, these histone cores, can be tightly compacted together, or they can be spaced out like beads on a string here in the open chromatin. So modifications to the histone proteins or to the DNA itself, and these modifications happen at cysteine bases in the DNA backbone, can regulate the accessibility. And cysteine methylation, seen over here in these red marks, is characterized of this condensed, silenced heterochromatin. So this DNA methylation is made by enzymes called DNA methyltransferases, and this is what you find in inactive or silenced genes. And until fairly recently, it was thought that this methylated silenced DNA was a permanent state until enzymes called the TET enzymes were discovered about 10 years ago and can actually demethylate this DNA and convert it back to an open active gene expression. So TET enzymes take this 5-methylcytosine mark and oxidize it to 5-hydroxymethylcytosine. So this 5-HMC is an epigenetic mark that we find in open and active chromatin. And we have found that this interplay between DNA methylation and methylation and demethylation seems to be what allows smooth muscle cells some of this ability to switch gene programs when needed. And we became interested in this idea because TET proteins, which were discovered again about 10 years ago, were found to be really critical in stem cells. So they've been found to be responsible for the property of self-renewal in embryonic stem cells, and also responsible for appropriate and effective differentiation of hematopoietic stem cells into all the different hematopoietic lineages. And interestingly, mutations in TET2 are the most common cause of myeloid malignancies. So we hypothesized that TET enzymes may contribute to this smooth muscle plasticity. And this was the work of Renjing Liu, a former postdoc in my lab. We published it in Circulation about 10 years ago. And she found that TET2, there are 3 of these TET family enzymes. She found that TET2 was expressed at very high levels in smooth muscle cells, higher even than what we see in embryonic stem cells. And when she looked at normal healthy blood vessels, so the red staining here is staining for a muscle protein in the arterial wall of a mouse artery. And here you can see that all of the cells in this arterial wall stained in green are expressing TET2. So TET2 is expressed in the healthy arteries, but in a model of intimal hyperplasia, here the letter N shows where the neointima is, and you can see it over here in this merged image. We essentially see no TET2 staining in these fibroblast-like hyperproliferative smooth muscle cells. We found this same relationship in human atherosclerosis. So this was a biobank of samples from George Toledas here at Yale from donor hearts that were not used for transplant. And he found what we found in looking at his graded samples is that TET2 was expressed at high levels in healthy human coronaries, but with increasing severity of atherosclerosis, the TET2 level dropped to almost nothing. And we did genetic experiments in our mouse model of intimal hyperplasia. And if we knocked down TET2 locally in a mouse artery and then subjected it to an angioplasty type injury, you can see that loss of TET2 greatly worsens intimal hyperplasia. But overexpressing TET2, so if we prevent it from being downregulated, we can prevent the intimal hyperplasia from forming. So TET2 is clearly a very powerful regulator of smooth muscle phenotype. And through a whole series of experiments in vitro and in vivo, we found that TET2 is in fact a master regulator of smooth muscle plasticity. It lies upstream of many of the master transcription factors. It regulates the contractile gene promoters. So when TET2 is active, TET2 is repressed by vascular injury and growth factors. But when it's active, it makes this 5-HMC mark in the DNA of the contractile genes that are associated with a healthy phenotype. And it drives expression of the pro-differentiation transcription factors. Whereas at this synthetic phenotype, or these fibroblast type genes, we find that TET2 is associated with a repressed chromatin state. So these genes are actually methylated and silenced when TET2 is active. We also find that there were different histone modifications that we could detect. There were further activating modifications at the contractile genes and repressive modifications at these fibroblast type genes in the presence of TET2 activity. So in ongoing work in our lab, we're looking at the idea of how TET2 interacts with other chromatin modifying machinery to globally promote a more open and active state at these smooth muscle specific genes while repressing the fibroblast type genes and other alternate programs of gene expression in smooth muscle cells. So we wanted to look and see whether TET2 might also have a role in transplant vasculopathy, and this is the work of Alison Ostreicher, a very talented postdoc. She was a PhD, but an incredible rodent microsurgeon, and I'll show you some of the neat surgery she was able to do in mouse models. So as you know, coronary allograft vasculopathy consists of concentric luminal stenosis of the vessels of the transplanted organ, and this occurs in a diffuse manner throughout the whole vascular tree of the transplanted organ. So unlike the focal lesions of atherosclerosis that are very localized and amenable to treatment with PCI, because this intimal hyperplasia is everywhere, it's much more challenging to treat. It's the leading cause of failure of transplanted organs. It occurs at a rate of 5% of heart transplants per year, and approximately 1/3 of heart transplant recipients will have this vasculopathy in 5 years. And there's an unfortunate lack of good treatment options. As I understand, the only Options at the moment are to increase or change the immunosuppressive regimen or to retransplant. And here you can see some examples of really severe occlusive lesions after heart transplant. So the model of how we think this process works is there is damage to the lining of the vessels of the transplanted organ. So the endothelium can be damaged during the procurement process. There can be ischemia reperfusion injury. There also are alloantibodies generated in the recipient that recognize the lining of these donor blood vessels as foreign. These antibodies recruit other cells like T cells. The T cells infiltrate the vessel wall where they secrete large amounts of cytokines, especially interferon gamma. And interferon gamma has been shown to be sufficient to drive the transplant vasculopathy response in mouse models. The smooth muscle cells then, in response to this immune injury, unlike the mechanical injury in angioplasty, this is an ongoing chronic immune injury to the smooth muscle cells, can promote them to go into this fibroblast type hyperproliferative repair mode, and you form these intimal hyperplastic lesions. So we hypothesize that downregulation of TET2 might also play a role in this pathology. So a simplified animal model that we use in the lab is a aorta graft, an aorta interposition graft. So we could take a segment of thoracic aorta from one mouse and transplant it into the abdominal aorta of a recipient mouse. And we can alter the background strains between these two mice. And the more different the strains are, the greater the mismatch, the greater the vasculopathy. Or we can use the same background strain when we want to look at a knockout model, and we can transplant a male aorta into a female recipient. And these Y chromosome antigens are sufficient to create a minor antigen mismatch and promote the vasculopathy. This is a time course after aorta graft model with a minor mismatch that Allison did. And here in brown, you can see staining for the T cell infiltration. And here's what the healthy donor aorta looks like. About 7 days afterward, you can start to see some T cells. And at 14 days, these T cell infiltration is really robust, where the space between the elastic lamina of the vessel is actually expanding from all the T cell infiltration. And you can start to see a neointima forming. And this continues to progress at 21 and 28 days post-transplant. And here you can see a pretty good size neointima that's forming. We look at interferon gamma signaling, and we can see that there's a strong interferon gamma signal at 14 days when there are a lot of these T cells. We can also see that TET2, this brown-red staining that was present at high levels in all the nuclei of the healthy donor vessel, is progressively downregulated, especially when there are a lot of T cells. And we see the same pattern with this 5-HMC genetic mark, epigenetic mark in the DNA, gets strongly repressed when with the T cell infiltration. We see the same pattern of TET2 repression in human coronary allograft vasculopathy. So these are autopsy specimens provided to us by Dr. Anita Huttner in pathology. And this is from a donor who died of non-cardiac causes. This is a relatively healthy coronary artery, and you can see the dark brown staining of the very elongated circumferentially arrayed nuclei of the smooth muscle cells in the healthy vessel. They all stain for TET2, whereas in the transplant vasculopathy artery, we can see the TET2 staining is greatly downregulated. You can also see this when you look at the 5-HMC epigenetic mark. Here's the healthy artery, and it's greatly downregulated in the transplant vasculopathy. We were able to quantitate this in a small biobank of human transplant patients, and we saw a very reproducible and significant reduction in TET2 expression and the 5-HMC mark with allograft vasculopathy. We were also able to see that this T cell derived interferon gamma seems to be sufficient to repress TET2 expression. So when we treated human smooth muscle cells in culture with interferon gamma, we saw a dramatic reduction in TET2 expression and activity. So we next asked what would happen if we were to transplant a graft that lacked TET2. So we made a smooth muscle specific knockout that lacks the TET2 gene, and we transplant it into a wild type host. And in this experiment, the smooth muscle cells of the graft are labeled, permanently labeled in green with GFB. And when we transplant a wild type artery, again, there's a minor mismatch. So there is some neointima that's formed here that's stained in white, but you can see there's a pretty good staining for the smooth muscle layer of the graft in green. We were surprised to find when we transplanted the TET2 knockout vessels that the green cells were greatly reduced, suggesting that we had a lot of the TET2 knockout cells in the graft were dying and disappearing. Despite the fact that these TET2 knockout cells were dying, we still had a pretty robust neointima forming, shown here in white, that was slightly larger than the wild type graft. So a lot of the TET2 cells die in the graft, but not all of them. So this experiment was a little puzzling to us at first. And so we looked in detail, kinetically, at what was going on here. And in the green, you can see the 5-HMC progressively decreases over time. What we found, and that's shown here in the green line in the graph, what we found is at the same time that the 5-HMC in TET2 was decreasing, both cell death and cell proliferation were increasing with superimposable curves. This was really surprising to us. Here in white, you can see this is a marker for apoptotic dying cells. And in the next line, you can see in pink is the proliferating cells. And there's a nice correlation suggesting that proliferation and death are closely related. So when we looked with high resolution here, we were able to see in white that dying cells are almost always next to a proliferating cell, shown in red. And this matched with the work that was published from another group who showed that in the setting of transplant vasculopathy, when smooth muscle cells die, they release chemotactic factors, including SDF1 alpha, that promotes the proliferation and migration of their surviving neighbors. And we found that this was also true in the TET2 knockout smooth muscle cells. So we saw smooth muscle cells lacking TET2 accelerate this whole process. You have more cells dying. They're releasing these factors that cause the growth in their neighbors. So we see T cells come in, they release interferon, TET2 is repressed, cells start to die. Some of them, you can think of it as the dying cells are spilling their contents that are affecting their neighboring cells. The neighboring cells are in turn hyperproliferating and forming these lesions. So how does understanding this mechanism, what does this tell us about potential implications for therapy? This brought us to the idea of looking at the effects of vitamin C in transplant vasculopathy. So vitamin C is actually an essential cofactor for this oxidation reaction carried out by TET enzymes. And there was a paper in Nature in 2013 that showed that treating embryonic stem cells with vitamin C dramatically increased TET activity in the cells. Here, these are embryonic stem cells. These are untreated. For treatment with vitamin C, you can see the bright red staining for the 5-HMC epigenetic mark made by TET enzymes. And here is an in vitro biochemical reaction. You can also see the profound increase in TET2 activity when these cells were treated with vitamin C. So these experiments and understanding the roles that TET enzymes play in cell differentiation led to experiments in mouse tumor models. And I mentioned that TET2 is one of the main mutations that causes leukemias in myeloid cells. And so in this study, they were able to restore the activity of TET2 mutant cells, not completely TET2 deficient cells, but TET2 mutant cells that still retain some but reduced activity. You could boost that activity with vitamin C. They saw anti-leukemic effects. There was another model with solid tumors where they were actually to show effects of ascorbic acid as well. And so this has renewed interest in the idea of high-dose intravenous ascorbic acid therapy for human cancers. So we decided to see whether we could boost TET2 activity in our transplant model. What effect would that have on the pathology? So here again is our model where the smooth muscle cell layer is marked in green. And these are control vessels that express TET2. When we transplant a mismatched artery, again in the control, this is the placebo saline situation, we can see that almost all of the green labeled smooth muscle cells are lost, and there's a sizable neointima that's formed. When we treated these mice with vitamin C, so we injected an ascorbic acid solution intraperitoneally, 4 grams per kg every day, starting at day 3 post-transplant. We did this for 1 month. I hope you can appreciate there was a really dramatic recovery of the smooth muscle cell layer. So treating these mice with vitamin C immediately post-transplant was able to prevent the smooth muscle cell death in response to the immune attack. And we have a small neointima forming, but it's much smaller than what we saw in the placebo control treated mice. When we did the same experiment in TET2 knockout mice, we saw that the vitamin C treatment had no effect at all. And in fact, we see a very large neointima that's formed here. And these are the quantities. When we look at the intima thickening that happened in the wild type mice, this was dramatically reduced by vitamin C, but no effect in the mice lacking TET2. Similarly, we were able to rescue the medial cells with vitamin C in the wild type, but no effect in TET2. So this suggests to us that even though vitamin C could have systemic antioxidant effects, could have effects on blood cells, immune cells, we think that the main target, therapeutic target in this scenario is really on the smooth muscle cells themselves, because in the smooth muscle specific deletion of TET2, there's no therapeutic benefit of vitamin C at all. So we were really excited about this, that we may have a mechanism that provides a rationale for how and why vitamin C might be beneficial in preventing transplant vasculopathy. By augmenting the activity of this crucial TET2 enzyme that limits the ability of the cells to switch into this repair mode, we may be able to dramatically reduce intimal hyperplasia. So we were wondering if there was any clinical evidence for this, and I was excited to find this study from Lancet 2002. This was a 1-year study of oral delivery of vitamin C and E after transplant, looking at transplant vasculopathy. And this was done by a group at the Brigham and at Oregon. So they started with 110 transplant patients. They were able to randomize 40 for the trial, about 20 in each arm. 20 got vitamin C and E together, oral doses, and 21 got placebo. And then 1 year later, they did IVUS to follow up on the transplant vasculopathy. They found no significant differences in lipids with the vitamin treatment, no significant differences in endothelial or vasomotor function. But when they looked at neointima progression by IVUS, they did see an encouraging significant difference. So the placebo group, the neointima increased by 8%, but the vitamin treated group had no significant change. It was at 0.8%. So I don't normally look at this type of IVUS data, but I put up the actual figure from the paper here if any of you are interested in the raw data. But I thought that this was encouraging that they saw these differences in year 1. And so this group then published 10-year follow-up data, which is also encouraging. So this is their follow-up study in the Journal of Heart and Lung Transplantation. And they saw a significant effect in their primary composite endpoint, which included diagnosis of CAV, PCI, acute MI, and death due to CAV. And their composite endpoint, they had a more than twofold increase in the placebo group compared to the vitamin C and E group. So again, only 20 patients, but pretty encouraging. I don't believe the secondary endpoint technically met statistical significance, but there were some very encouraging trends there. So time to onset of the vasculopathy was 7.5 years with the vitamins versus 4.5 years with the placebo. There was no difference in time to PCI. And so the reason they may not have had significance is because of the way the study was done. It was very small. After the first year where they saw the benefits with the 500 milligram dose, they doubled the dose of both vitamins, but more importantly, they had significant patient crossover. When they saw the results of the 1-year trial, a lot of the patients switched over to the drug treatment group. That may have made it much harder to detect a significant difference in some of the endpoints because they had a lot fewer patients in the placebo group. But they did note a delay in CAV progression. Despite these encouraging results, post-transplant use of antioxidants is not routine, and this study is too small to draw generalized conclusions. But paired with our new mechanistic data that suggests a reason why vitamin C might be beneficial, this highlights a need for further trials. So I've never looked at vitamin E. I don't know that there's any effect of vitamin E on TET enzymes, but so I don't know the effect from these clinical studies of the contributions of vitamin C versus E versus a combinatorial effect. But we thought that this was encouraging. So we're hoping that larger studies might support this approach, and I'm very excited that Lavi has agreed to help out with this and has brought in her collaborator, Dr. Monica Colvin from University of Michigan, to try to see if we can do a retrospective analysis of transplant cohorts, those who routinely receive vitamin therapy versus those who do not, and see if we can find any evidence for delay in onset or reduction in severity of allograft vasculopathy. So this is ongoing work, but I'm kind of excited that our mechanistic studies suggest a reason to revisit this therapy. The fact that this therapy is safe, inexpensive, easily accessible, makes me really excited that this may be something that we could do with relatively low risk that might help outcomes for transplant patients. So I just quickly wanted to show you another model that we use in the lab. I hope you might enjoy seeing this. So our aortic graft model is a very simplified model, but we can also do this heterotopic heart transplant model that better models actually coronary artery vasculopathy. So we're not sophisticated enough yet to replace the mouse native heart, but we can add a second heart to the abdominal circulation, and we can, again, vary the mismatch of this heart to manipulate the degree and the timing of the vasculopathy. And so this heart is attached, the aorta of the second heart is attached to the abdominal aorta of the host. And the heart is perfused retrograde through the left atrium, out through the left ventricle, and into the inferior vena cava. But the good thing about this model is because of the origin of the coronary arteries on the aorta, they are perfused in the proper direction. And so we can look at blood flow and coronary allograft vasculopathy in these whole hearts. These hearts continue to beat. We can use our genetic lineage tracing that I alluded to. So here's an example of a cross-section through the heart where the coronary arteries, you can see the smooth muscle cells have this fluorescent green labeling. And here's a close-up of this coronary artery where you can see the green smooth muscle cells surrounded by the sea of red cardiomyocytes. So we can actually enzymatically digest these mouse hearts into single cells and recover just the smooth muscle cells and look at what's happening. So we can do our sections, we can do histopathology on the hearts, we can also do RNA sequencing, genome-wide epigenetics, things like that, and the isolated green smooth muscle cells. And this is a movie of what this heart transplant looks like. This is the transplanted heart nestled among the intestines of the recipient mouse. And here, this is a same strain transplant, so no vasculopathy versus a strain mismatch. And you can see the differences in the heart rate and beating pattern, where the non-mismatched heart is beating much faster, it's beating rhythmically, and it's pretty well perfused. Whereas the mismatched heart, you can see it's beating more slowly, more irregularly, and there are these gray areas where it's more poorly perfused. The other nice thing about this system is you can actually palpate the second heart right through the abdomen. It's very close to the surface, so you can feel the heart rate, and you can monitor when it's slowing down, when it may be getting close to failure, so you know when to harvest the experiment. So this is another exciting technique that we have in our arsenal. This is another example of this GFP labeling of the smooth muscle cells. And if this is one of these nice time courses that Allison did, and if you look at this yellow arrow here, we can see a single green smooth muscle cell entering the lumen at 7 days post-transplant. And we were really excited that we were able to capture this, because that has allowed us to do single cell RNA sequencing. So as I mentioned, we can dissociate these into single cells and sequence the RNA in each cell individually. And we're looking to see if we can identify gene signatures in these single cells that are the first ones to go out into the lumen, to see if we can understand what are the unique molecular events in those cells that are causing them to enter the lumen and initiate this pathology. So these are ongoing studies in our lab that we hope will provide us even more insights about the pathology of this process and ways that we can intervene early after transplant to reduce transplant vasculopathy. So to summarize, we've identified TET2 as a novel epigenetic regulator of smooth muscle phenotypic plasticity. It's repressed in pathologies, including transplant vasculopathy, and promotes smooth muscle cell death and proliferation. We've shown that augmenting TET2 activity with vitamin C may have therapeutic potential, and we hope that we may be able to target these pioneer cells that drive the lesions. So these are my team members who made all of this possible. Especially Allison Ostreicher, who did the bulk of the work on this transplant project, and Renjing Liu, who made the initial discoveries about TET2. Thanks to all my Yale collaborators, including John Hua and Lavi. I'm so excited to be collaborating on the ongoing clinical implications of this. And I'd be really happy to hear your thoughts. Any ideas or suggestions for us would be greatly appreciated.
Thank you so much, Dr. Martin. Lavi, do you have anything that you'd like to mention before we see if there's any questions?
Kathy, that was an excellent talk. Thank you so much. Just had a quick question. I know it's with timeline, the mice don't live a whole lot of time, but if you introduce ascorbic acid a little bit later in the timeline, would you see the same effect on TET2 and vasculopathy changes? I mean, and then number two follow-up is, would modifying immunosuppression with induction therapy, especially with agents such as T-cell depleting agents, such as thymoglobulin, which has a protective effect on ischemia or perfusion injury and obviously depletes the T cells, and then the downstream pathway of interferon gamma, would that have an impact? I'm not sure if you had a chance to look at that in the mice model.
Yeah, great questions. Thank you. So we haven't tried delayed treatment with vitamin C, but that would be important. I think the thing to note about our mouse model is it's kind of accelerated compared to what happens in the human pathology. So we're seeing in 1 month what might take more than a year to happen in a human. And I think we're seeing the effects of vitamin see when we treat early because the TET2 has not yet been downregulated. I'm not sure how durable this response will be in the mice if the TET2 is eventually strongly repressed. I'm encouraged because in the humans, I think it probably takes a lot longer to downregulate TET2. And so as long as there is some TET2 present, vitamin C should have some effect, even if we're coming in at a later time point. Whether vitamin C has any effect on TET2 expression and can prevent the downregulation, we haven't looked at that and we don't really know. But that would be important, I think, to see how long lasting the effects might be. The question about immunosuppression is a good one too, because I think it is that the T cells and the interferon that are repressing the TET2. And so if we can, anything we can do to limit those T cell responses should be helpful. And others have shown that anything that reduces the amount of interferon can reduce this in the mouse model. I didn't go into it today for the sake of time and simplicity, but we've shown that rapamycin, the drug-eluting stent agent and an immunosuppressant, increases expression of TET2. And so an immunosuppression regimen that includes rapamycin would probably be synergistic with the vitamin C, because it would be increasing the expression of TET2 at the same time that vitamin C would be increasing the activity of TET2. So those things might work quite nicely together.
Thank you so much.
It's very exciting. Anybody else have any questions for Dr. Martin? I just had one thing, if nobody else does. It's more kind of in relation to your proposed trial design. Like when you're doing a trial like this, where you know, you're looking at nutritional supplementation, that, you know, there could be a lot of confounding with you know, just regular dietary practices or recall and things like that. How do you go about ensuring that you're getting accurate data in that type of way?
The clinical studies actually measured the vitamin levels in the patient plasma. So that's one way to see what level they're actually taking. I think they excluded, so they had, you notice they excluded quite a lot of patients in that initial stage of the trial. It may have been that some of them were already taking vitamins, and so that would exclude them. I agree, there's all kinds of differences in dietary intake of vitamins or taking supplements that could impact this, but that would have to be factored, definitely factored into any study.
Yeah, yeah. Well, thank you again, Dr. Martin, for taking the time, and I'm very excited to see what yourself and Dr. Belancanda and Dr. Hua do with this great work, and hopefully make a world of difference for all these patients in the future.
Well, thank you for having me. And if anyone has any thoughts in the future, please reach out to me or Lavi. We'd love to hear.
All right, thanks so much.
Thank you.